Start. So good afternoon, everyone. Welcome to this NITheCS Quantum Research Group at UKZN webinar. This afternoon, we are really very fortunate to have Dr. Ismael Galván. I hope I pronounce it correctly. I'm not sure what the meaning.
Yes.
Yeah.
No, it's Galván, yes.
Yeah, of the accent is. Yeah. And as you saw in the announcement, he's speaking about the quantum basis of organic evolution, which is really an exciting topic that is close to our hearts. Yeah? And Ismael studied biology at the Complutense University in Madrid, in Spain, and spent some time as postdoc at University of Paris Sud, at University of Lisbon, and at the Doñana Biological Station in Spain, probably Madrid, I'm not sure.
No, in the south of Spain, it's in Seville.
In Seville. Okay, okay, nice. Also nice. Also nice. Very nice. Sorry, I should have made my homework. And now he's a staff scientist at the, which is also a very nice location, at the National Museum of Natural Sciences in Madrid. Yeah? And he's always been interested in the interface between chemistry and biology and physics. Yeah, if I can add that. Yeah. And today he will share with us his thoughts about why he thinks that evolution is a quantum thing. Yeah. So Ismael, we are really very grateful that you took the time and the trouble to offer us a webinar in South Africa. Yeah. And if you would like to start sharing your screen, you're welcome to start your presentation. And Ismael, sorry, we set up this webinar as a seminar, so everybody has the right to speak at any time in principle. Are you happy if people ask questions when they arise, or would you like us to wait until the end of your talk?
I'd be more than happy for that. Yes.
Okay, okay, perfect, perfect. No, just just to make sure that we have the games of the rules set before we start. Okay, fantastic, Ismael. Then, if you would like to start, you're most welcome.
So thank you very much for your invitation, and I would be delighted to go there and give a seminar in person when things become normal again.
We'll do it next year.
I hope so. So let me share with you this presentation. First of all, I'd like to thank you again for your interest in this strange thing, which is the interface between physics, chemistry, and biology. That I know it's a little odd for, especially for a biologist like me. But I have, as you said before, I have always been interested in the interface between biology and physics. And I have always understood that biology doesn't exist because it's just a combination of different processes in the chemical and physical sciences. So these ideas that I will show up here are approximately the result of my particular view of biology that is not shared by many, but hope you show some interest, and we can discuss this further in order to get deeper insight in this phenomena. I'd like to start mentioning Antonio Lima de Faria and his work. Antonio Lima de Faria is still a geneticist from Portugal, but based in Sweden and the University of Lund. And he was a pioneer in suggesting that many macroscopic properties of organisms may have an atomic imprint from the mineral world. he didn't know when he published his paper in 1997 in the 90s. He didn't know very well how to express his thoughts, but he was a pioneer in these ideas. Especially because I think that he said one important point, which is the opposite to the generally accepted view today about biology, about biological forms, because it is generally accepted that all biological forms are new, but actually no biological form is essentially new, because the rise of biological forms is an increase in complexity, which is made by building on simpler forms that had previously evolved and selected. Indeed, some not very old works like this one in Nature from 2003 confirm that biology grows like combination of systems on different levels, from the simpler to the more complex levels. What this means is that every level of evolution is dependent on the previous one, and everything in biology comes from the most basic level, which is the mineral world. And this is an obvious thing, but for me it's important to highlight this fact that biomolecules are the building blocks of organisms, and biomolecules are made up of atoms that are taken from the mineral world. So organisms are made of atoms taken from the mineral world, and minerals are one step earlier than organisms in complexity. This is also obvious, but it is also known that despite that fact, some inorganic compounds can produce, in some cases, organic molecules. Many minerals containing iron can produce, under exposure to ultraviolet light, can fix the carbon dioxide and produce small organic molecules like formaldehyde. And also dissolved mineral salts of iron, and producing the oxidation of iron can promote the reduction of nitrates and nitrites to ammonia, and this in turn can produce the change of a small molecule of like pyruvate to the amino acid alanine. These simple steps are very important because they are the core of the origin of life. They are the core of the origin of life, and they show an obvious connection between the mineral and the organic world. And it is this connection what brings the source atoms of biomolecules. So it is expected that minerals leave an atomic imprint on organisms. So many important macroscopic characteristics of organisms are actually an atomic imprint taken from the mineral world. This has never been told, or this has never been suggested before the work of Antonio Lima de Faria, but it is, I think, actually a possibility that may be very important to understand how organisms are and why are like they are. So this atomic imprint from the mineral world in organisms may be responsible for some physical-chemical properties of organisms, which has only been envisaged by some author before. This atomic imprint and the atomic properties in organisms may be transferred, may represent a transfer of information that could be carried across complexity levels, and thus affect the form and function of organisms, affecting the macroscopic characteristics of these organisms, and in this way representing an evolutionary source of evolutionary innovations. So it is possible that many differences that we see in organisms are actually the result of a different atomic imprint in them that has nothing to do with the DNA. Indeed, genes do not originate the form and function of organisms. The genes only fix some already existing variants of form and function and promote their repetition with time. But genes do not create anything new. And an example, I'd like to highlight the shell of mollusks, which is made of calcium carbonate. It's a simple molecule, but it is known that it is the disposition of atoms in the molecule of the calcium carbonate which is responsible for the basic shape of the shell, while genes only determine the combination of the molecules into keratin matrices, and this only creates differences in some specific properties of the shell, like the size and sphericity. So it is the atomic disposition in the molecule what determine the more relevant properties in the shell, which is the basic shape of the shell. So this atomic imprint in organisms may be transferred when molecules in organisms are synthesized, may be transferred when a new molecule is synthesized inside an organism, and may be transferred in this way across generations. So it would be the process of molecular biosynthesis responsible for the transference, for the transfer of this information across molecules and across generations. Nucleic acids are the molecules of heredity, and they determine the characteristics of the molecules, of the new molecules that are synthesized inside organisms. This is made by genes. But there is no code determining which kind of atoms form part of biomolecules. So this information must necessarily be taken from the minerals, which is the source of atoms. So such an information, such an atomic imprint in organisms may represent a DNA independent mechanism of inheritance that has never been investigated, and may represent a source of important macroscopic characteristics in organisms. But which information, which is specifically that atomic information, that atomic imprint on organisms, what could be the content of such information? I think that one important point here is the existence of stable isotopes, because not all, as you all know, not all atoms are equal. they differ. The atoms of the same element differ in the number of neutrons, generating isotopes. And isotopes have their origin with the universe. In the case of the 5 most common biological elements, they exist in 13 different stable isotopes, which creates a great variability in organisms in the form of biomolecules with these isotopes, because the isotopes are combined in different proportions. So no all biomolecules are the same; they differ in the isotopes. I am showing only the 5 most common biological elements, but of course there are others very important elements for organisms, such as some metals that act as cofactors in many biochemical reactions that always are present in different isotopes. Which is different in different proportions, so it is relevant too for organisms. But at least the 5 most common elements in biology at least exist in 13 stable isotopes, and this is the basic variation of biomolecules. And why is this important? This is important because before the 60s, it was believed that the electrons or nuclei are the only responsible for the course of chemical reactions. But in the 60s, it was evident that isotopologues, which is the name that receives 2 molecules, 2 chemically identical molecules that differ in the isotopes that they bear, that isotopologues behave differentially in biochemical reactions. So this was evident, and it was soon realized that the different masses of isotopes, because isotopes with a higher number of neutrons have a higher mass, have a higher volume, and the isotopes with an odd number of neutrons have a magnetic moment, they affect the course of the chemical reaction. And it is this effect of isotopes on chemical reaction which creates an isotope fractionation in nature, because isotopes are not randomly distributed in nature. They experience a series of influence in the biosphere during different abiotic processes that occur in the biosphere, and due to the properties of the isotopes, they get fractionated in different proportions, and then as a consequence of the biosynthesis in plants, they also get fractionated in this process of biosynthesis. And lastly, as a consequence of the chemical reaction of metabolism in animals. And this is important because we now know that many metabolic reactions in plants and animals are mediated by quantum processes. So animals that share the same metabolism, the same chemical reaction of metabolisms, are expected or should be expected to have a similar isotopic composition in their constituent biomolecules. If there is an effect of quantum processes in these metabolic reactions, animals that share the same metabolism should be also a similar performance of quantum processes in those biochemical reactions. So the key point here is that animals of the same species have a similar isotopic composition. currently there are two bodies of evidence of that fact. The first body of evidence comes from the isotope resonance hypothesis, which shows that there is an optimal an optimal proportion of isotopes under which chemical reaction take place and accelerate. And this is probably related to the fact that organisms in different experiments show a resistance. Animals, for example, that are grown in different isotopic composition, in isotopic composition that is different to that of their body, they show a resistance to change the isotopic composition of their body. And this is probably related to the isotope resonance hypothesis, because there is organisms do not have a random proportion of isotopes in their biomolecules, but there is an optimal proportion under which the metabolic reaction takes place. And this could even explain why the genetic code was selected to have only a particular number of amino acids, and why it is possible to create many combinations of 3 peptides. And this is probably related to the fact that only certain compositions work well in biology. And the second body of evidence that animals may have a species-specific isotopic composition comes from studies of the isospaces. In ecology, an isospace is just the relationship between the proportion of two different isotopes in their tissues. When we plot the isospaces of different species that are very similar ecologically and morphologically, it is clearly seen that even with this similarity, the isospaces of the different species do not overlap. And even in humans, comparing humans with Neanderthals in hominids, they are obviously very similar genetically and morphologically. The isospaces of these species of modern humans and Neanderthals do not overlap. So this all is suggesting that species have a specific isotopic composition, and if they have a specific isotopic compositions in their biomolecules, they may share a similar performance of the biochemical reaction of metabolism, and this may be giving rise to evolutionary origins. This is basically the hypothesis that I have and that I wanted to share with you, and I'm summarizing here in this graph, because isotopes from the mineral world are fractionated during abiotic processes due to the properties of the isotopes. Then during the metabolic, the chemical reaction of photosynthesis in plants, and lastly, during the chemical reaction of metabolism in animals. It is here where quantum processes start affecting, because we know that many metabolic reactions are actually quantum processes. So if different species have different metabolisms, each species may have a specific isotopic profile. And we also know that isotopes have an influence on the performance of quantum processes. So species different in the isotopic profile may have a different performance of the quantum processes. If a given trait depend on the performance of quantum processes, then the isotopic composition of the different species may be responsible to determine the rise or the limitation to rise of that specific trait, because it is the isotopic composition what would be limiting the quantum processes that control the metabolism, that in turn control the rise of the trait. But which metabolic reactions are mediated by quantum processes? You know that very well, but I will only express two examples here because they serve as examples to potential explanation for the evolutionary origin of two specific traits. In the case of magnetoreception or magnetodetection, this could be an explanation for the evolutionary origin of migration ability, the ability to perform long distance migration. And in the case of enzymatic reactions, there could be an explanation for the evolution of flight, because flight requires an extremely high energetic metabolism that could be provided only by a highly performed enzymatic reactions in the energy metabolism. So I will start commenting on the case of the magnetoreception as a potential explanation for the evolution of migratory ability. Many animals perform long-distance migration, and it has always been a mystery how they orientate during navigation around the world. But it is evident that they are sensitive in some way to the magnetic field of Earth. Recently, it has been shown that in a bird, the robin, the European robin, cryptochrome 4, which is a flavoprotein, which is the only class of protein which forms radical pairs upon photoexcitation. Cryptochrome 4 was present in the retina of this bird, and the protein, the cryptochrome, bears a flavin radical and a tryptophan radicals that may produce a radical pair. I know that you know this very well, but I will summarize the radical pair as a mechanism in which a molecule gets photolysed, produce two radicals that before diffusing apart into the bulk solution, they form an intermediate state, short life state, that is called the radical pair. The radical pair to the chemical bond between the radicals can be regenerated if the sum of the spins of the radicals is not it's zero, but it is known that the radical pairs in the beginning are formed they are in the singlet electronic state with anti-parallel spins, but they are soon converted into the triplet state, and it is magnetic forces like the electronic Feynman interaction and the electric nuclear Fermi interaction and microwaves which promote the conversion into the singlet state. And this is important because only the radical pairs that are converted are converted back into the singlet state can regenerate the starting molecule. And for birds bearing cryptochrome in the retina, this is the basis for the magnetic orientation, because it is the recombination, specifically the disruption in this recombination, what provides them with an information about the magnetic field, because the recombination is sensitive to the orientation of the magnetic field. So this provides them with quantum information that is useful to orientate during navigation. Presently, it has experimentally shown that in working with pigeons, that cryptochrome 4 actually forms radical pairs in the retina. For example, the prediction for a radical pair is that the correlation, the spin correlation of electrons persists for more than 1 microsecond, and the transient electromagnetic, the paramagnetic resonance spectrum show absorptive and emissive peaks that are around that value, around one microsecond. So this article represents a body of evidence of the actual formation of radical pairs in the retina of birds. So the radical pair mechanism is actually working in birds. And why are isotopes important here? Isotopes are important here because the influence, one of the stronger influences that isotopes affect on chemical reactions are in the radical pair mechanism. I am copying here an example shown by the seminal paper by Wychachenko in 2001 with the molecule of dibenzyl ketone, which shows that the molecule under exposure to ultraviolet light forms 2 radicals and forms a radical pair, which is in the triplet electronic disposition state. And the reconstitution of the starting molecule depends on which isotope the radicals bear, because if it bears carbon-13, the heavy isotope, there is a fast triplet singlet conversion and establishment of the reestablishment of the starting molecule, because there is a hyperfine coping between the amperic electron and the magnetic nucleus of the carbon-13. But instead, if it bears carbon-12, only protons contribute to the hyperfine coping. So the recombination, the triplet singlet conversion is strongly delayed, and recombination does not take place or does not predominate. So the photoproducts give rise instead. So in the case of birds with cryptochrome 4 that have 2 radicals, 1 radical of flavin and 1 radical of tryptophan, it would be important to see if the species differ in the isotopic composition of these radicals, because an isotopic composition rich in isotopes with an even number of neutrons would impair the triplet singlet conversion and the recombination of the starting cryptochrome protein would not be possible. So these animals could not migrate, could not have the ability to orientate with a magnetic field, and would not be capacitated to, could not perform long-distance migration. So this could, the isotopic composition of the radicals and the protein cryptochrome 4 could represent an explanation for why migration has only evolved in some species and not in others. This is the hypothesis. The ability to conduct non-distant migration has evolved only in animals with a prevailing contribution of odd carbon and nitrogen isotopes to flavin and tryptophan in the cryptochrome molecule.
Please can I ask a question quickly? Sorry to interrupt you.
Sorry?
Please could I ask a question? Sorry to interrupt you.
Yeah, yes.
Are there enough of these isotopes naturally occurring to be taken up by other species?
This is the challenge. It is for me. It is. It would be necessary if you are asking if there are actually differences between species for the animals in the isotopes.
Or just do these isotopes occur enough naturally? And how abundant are isotopes of carbon and nitrogen?
Well, there is always a different proportion of these isotopes. We don't know the proportion of the different isotopes, but for example, in the case of cryptochrome, the carbon and the molecules always occur in some proportion of the carbon and nitrogen isotopes. So the question will be to see what is the exact proportion of the different isotopes, but they are always there. So the question is if the differences between species in the proportion of the isotopes are high enough to produce differences in the that are relevant to produce differences in performance in the radical pair mechanism. I don't know if that answers your question.
Thank you very much.
Yeah, so I just like to say that the challenge here would be that to make a theoretical calculation of what would be the effect of the different proportion of isotopes in the molecule of cryptochrome on the performance of the radical pair mechanism. This could be aborded from a theoretical point of view exclusively, but it would be also necessary to measure experimentally those differences in performance of the radical pair mechanism in the retina, in the eye of different species that differ in the isotopic proportion of cryptochrome 4. And lastly, I'd like to see that it is also open the question of how is the quantum information of the radical pair mechanism transferred to the retinal nervous system of birds. So this is a completely open question, because it is necessary to know how birds take the quantum bit is established in the cryptochrome and use this information to navigate. In general, this is a very relevant question for any interaction between the quantum processes and biology. But the second example is an example on enzymatic reactions. Enzymatic reactions are essential for life because the specific conformation of the enzyme and its substrate allows having a product by lowering the activation energy necessary to reach those products after producing a transition state in which the activation energy is lowered. This is the classical kinetics explanation. But as you all know, some enzymatic reactions have been shown to be actually quantum processes in which quantum tunneling is acting. So the quantum tunneling would be actually the responsible of lowering the activation energy in the enzyme substrate interaction. This is very interesting because isotopes, as you know, affect the course of enzymatic reactions. This is very well known from a classic kinetics view. But now that we know that quantum tunneling is indeed responsible for these reactions, it is possible that many explanations that have been tried to make with classic kinetics point of view are not sufficient to explain all the behaviors and enzymatic systems. For example, this recent study about the classical system of the enzyme luciferase, which oxidizes the protein luciferin, showed that a small concentration of heavy isotopes in the medium exerted disproportionately strong effects on the kinetics of the enzymatic process. This is also related to the isotope resonance hypothesis, and maybe it is the quantum tunneling explaining this pattern of influence. For me, this is important. This may be important to explain the evolutionary origin of flight, of powered flight. This is a very interesting question in biology because it has remained a mystery for decades, for centuries, indeed. Because we know that during the course of evolution, flight has only evolved 5 times, 4 if we exclude the flying fishes. And the interesting thing is that in all the cases in which flight has evolved, they have this trait has evolved from in a completely independent way from ancestors that did not fly. So flight is really a completely innovative evolutionary innovation. For example, in the case of bats evolved from non-flying mammals. So they, about I think 16 million years ago, so bats acquired the capacity to fly from zero, it was a complete invention. It was again Antonio Lima de Faria who suggested that maybe the animals that fly have some common atomic traits, without going beyond that, but only that suggestion was very interesting. And recent studies indeed may go in that direction, or at least show that the evolution of flight requires a high performance of the energy metabolism, because flight is a highly consuming activity. So only animals that have a high performance of the energy metabolism would be able to fly. When comparing bats with non-flying mammals, it was found that the difference in the genome was in genes that encoded enzymes that catalyze the oxidative phosphorylation process, which is the basic process of the energy metabolism. And when flightless birds and flying birds are compared, in the case of birds, flight has been evolutionarily lost more than 60 times. When comparing flightless and flying birds, it was found that exploring the genomes of the different species, it was found that there have been a selection in genes that encoding again enzymes of the energy metabolisms, in particular enzymes that catalyze reaction in the carbohydrates and lipids metabolism. There is an enzyme which is particularly important in the energy metabolism, which is cytochrome C oxidase, because this enzyme catalyzes the reduction of the oxygen molecule to water, releasing in the process half the total energy which is released during the energy metabolism. This enzyme catalyzes the transfer of electrons through a binuclear active site and the transfer of protons through the membranes of the mitochondria, because the enzyme is located in the inner mitochondrial membrane. And in particular, the nuclear active of the enzyme has a tyrosine residue, which is redox active. Is get reduced in every step of the enzyme, and the tyrosine is coupled to an M group. So the reduction of the enzyme is the basic functioning mechanism of the enzymatic process. That, as I say, is the key enzymatic process in the energy metabolism of vertebrates. In particular, there are 10 limiting energetic barriers for the enzyme. So if seeing the solution of the Redinger equation for quantum tunneling, it is clear that a particle mass is critical to tunneling. So if these limiting barriers, these limiting energetic barriers are overcome by tunneling, then it is expected that differences in the isotopic composition of the tyrosine residue of the enzyme could create differences in the performance of tunneling. So it is expected that animals that have evolved the capacity, the ability to fly, have a higher performance of quantum of tunneling, and this should be related to their isotopic composition. So the isotopic composition would provide the ability to fly only if the isotopic composition is has certain values. So again, here the hypothesis is that certain organisms with certain isotopic composition of the enzyme cytochrome C oxidase may have a high enzymatic efficiency that have promoted the appearance of flight. this may represent an explanation for the origin of flight, which, as I said, has remained a mystery until today. So the challenges again would be similar to the case of cryptochrome, which would be to calculate theoretically the effect of the different isotope composition, odd even isotopes proportions on quantum tunneling, and also to determine experimentally how would be the difference in quantum tunneling in different species that differ in the quantum in the isotopic composition. So these questions, I think that if solved, they may represent a non-adaptive and non-organic source of evolutionary innovations, which would be completely independent of the DNA. It also represents a connection between the mineral and organic world that I think has never been nor even suggested. And it may unveil attributes of organisms that are not carried by their genome, by their genes. So now it would be necessary to explore these challenges that I have suggested, but also to unveil other biological processes that are mediated by quantum processes. For example, I was thinking that the brain could be considered like a quantum machine. It could be calculated how much volume would be necessary for the quantum machine, because there is a large variation in brain size between animals. Even within mammals, there is a large variation in brain volume, in brain size, that is related to cognitive ability. So if that calculation could be done, it could be related to, it could be seen if the predictions fulfill the observed variation in brain size and cognitive abilities in animals. That would be, I think, a body of evidence that quantum processes are governing the development and functioning of the brain. If that would be combined with information about the isotopic variation in organisms, I think this could even open a completely new field that could be termed quantum evolutionary biology. So this is all I wanted to tell you. I hope you find these results interesting, and I'm open to any question from you if you have.
Yeah, Ismael, thank you very much. It was a fascinating talk, and it's really an exciting topic that that we were looking forward to work on with you. We had a question of Tiffany during your talk. Are there other questions from the participants? And please just unmute and ask your question.
Thank you, Ismael. It was really interesting, and I do hope you find some differences when you do this work. Yeah, I don't really have a question, but just maybe just the question that, so you will be then. Well, what I wanted to try to understand was, so normally like carbon-14 is used for dating of old samples, but you will be here looking at modern samples, and you will be looking in different tissues. Is that, do I understand it correctly?
Yeah, yes, I would be looking the hypothesis is that a given animal should be the same isotopic composition in all its biomolecules, in all the tissues, because there is variation in the isotopic composition of animal tissues due to diet. but there is a very small degree of variation due to diet, and indeed some experimental work shows that animals show resistance to change the isotopic composition of their bodies when given diet with different isotopic composition. So I would expect to find approximately the same isotopic composition in all biomolecules of a given animal, but that would be indeed tested. But to be more specific, I would search for in a specific molecule. In this case, in the case of when looking for differences between migratory and non-migratory species, I would look for in the cryptochrome 4 molecule of the retina specifically, and compare the isotopic composition of carbon and nitrogen isotopes in the protein and between species that differ in the capacity to migrate. And in the example of flight, I would look for in the cytochrome C oxidase, and specifically in the tyrosine residue of the enzyme, because it's responsible for the reduction of the tyrosine residue, responsible for the enzymatic action of the enzyme. So I expect to have the same isotopic composition in all biomolecules of a given animal, approximately. But to test these ideas, I would at least start measuring in the specific molecules.
Thank you. That's fascinating. I will look out for your papers.
Yes.
Thank you. And I see Thomas has a question, and then Bethany. Thomas, if you would like to start.
Yes, thank you, Francesco. Hello, and thank you very much, Ismael, for a very interesting talk.
Thank you.
I'm trying to understand your underlying paradigm. So you are saying that somehow we have the mineral world and we have the organic world, and somehow the mineral world gives some imprint onto the organic world that is kind of defining it in some way. So we have species that can fly because there is this enzymatic effectivity, and then you made the other example with the navigation, because we can sense the magnetic field very well. What else? I mean, if you think of all the different things that animals can do, would you say there is always some mineral or some mineral basis? There always a difference in the number of isotopes, or is that what you're looking for, or is this just a special case that you say, yeah, look, in this, here are 2 examples where this manifests, or do you have a more general statement?
Well, I'd say that I'm more generalist, but this is why I say that it is important to see what are the key point here is metabolism, because organisms work through metabolism, and metabolism is made of different chemical reactions. So here, the important thing here is to see what are the chemical reactions of metabolisms that are actually mediated by quantum processes. I know that everything is quantum, but in some specific examples, it seems that the control of quantum processes is particularly relevant, like in the case of plant photosynthesis, and the enzymes, and also the radical pair mechanism, which are the examples that we now know. But as you know, it has just started to be realized that many of the reactions that occur in organisms are mediated by quantum processes. So I will not be surprised if we see that many more processes than this that I have designed here are actually mediated by quantum processes. I would not be afraid of saying that maybe all the basic characteristics of organisms, the basic characteristics, know the fine-tuning traits. For example, differences in eye coloration could be well explained by genetic differences. But the evolutionary origin of the eye is a more complex thing. So I am referring to these basic things, and this is why I mentioned the example of the basic shape of the shell of mollusks, because the basic shape, which is the shape that we all know of the mollusk, it has to be with the deposition of atoms in the molecule of calcium carbonate, and not with how the molecule of calcium carbonate aggregates into keratin matrices. So it would be necessary to start by watching at specific processes like these examples. But I would say that all the basic characteristics of organisms could be explained through an imprint from the mineral world, which is the differences between organisms in the proportion of different isotopes in their biomolecules, and these differences in the proportion of isotopes in the biomolecules affect the course of chemical reactions in their bodies.
Chair, can I ask a follow-up question?
Yes,
sure.
yes, please.
Thank you very much. Yes, that's very interesting. But in a sense, are you then not saying that there is a static thing in evolution? So it's not, if I understand you correctly, you are introducing something that will never change. You say there's the mineral world, and we have certain minerals, and then you derive some characteristics from that, but we will not get away, we will not find more characteristics. I mean, in other words, I want to say not more characteristics will evolve. Is that what you're saying? The number of characteristics is somehow kind of limited?
No, because, for example, the evolution of flight, if the origin is actually related to the presence of some isotopic composition in some animals, it doesn't mean that with such isotopic composition, flight automatically evolves and remains the same during evolution. Because during evolution, there are also genes that get selected for in relation to the fitness characteristics that the results of those genes make. So genes repeat the same molecular combination. If the environmental conditions do not favor the course of the molecular combination, the trait would not evolve even if the isotopic composition of the animals give them the capacity to evolve the trait. So I think that the isotopic composition gives the animal the capacity to evolve the trait, but this does not necessarily mean that they will evolve the trait. It will other things more related to the genetics that will ultimately determine the chance to arise the trait. At least this is the general view that I have of these things.
Thank you. And I think we have one more question of Bethany. And please, if you are ready for the question, just raise your hands and we will sort it out. Bethany, please.
Thank you so much for a very interesting talk. I was just wondering, do you think brain volume is less important than metabolism, because brains are high energy metabolic organs? And has anyone looked at isotopic constitutions across different species brains? I'm asking because isotopes have been shown, externally introduced isotopes have been shown to have different effects in the brain, like lithium 6 and 7 for depression and xenon for anesthetic.
But specifically, what do you mean that? If you are asking if I think that the energetics of the brain, be more specific about your question.
Okay. Yes. So you said brain volume might be explained by this, but do you think brain volume is less important than the fact that more intelligent brains probably need higher energy metabolism? So then you've got the same thing happening with enzymes. And has anyone actually looked at isotopes across brain species? Because isotopes do play a role in brain function.
Well, higher brains require more energy, but I have worked a bit in making comparative analysis of brain size between different species, and as I know that there is an association between cognitive capacities and brain size. Of course, I am not talking about absolute brain size. I'm talking about relative brain size, relative to body size. Yeah. So I think that everything is related here. I was only suggesting the idea of brain size because of the idea of the space necessary to make a quantum machine working, considering that the brain was a quantum machine. But in relation to the enzymes, I don't know if there should be any relationship between the cytochrome C oxidase and brain size, because larger brains require more energy and then a higher performance of the energy. But when we consider the different organs and animals as a system, it is frequent to see compensation between tissues and between organs. So, for example, animals that have big brains have had to reduce others organs. So I would be. It requires more thinking. But for example, in humans, it is now assumed that the enlargement of the brain was only possible when the diet changed to a more carnivorous diet, and so the gut size diminished. So there is an obvious relationship between brain size, energetics, and the metabolism. But when thinking in the enzyme of the energy metabolism, yes, it would be necessary to control, when comparing species, for these others mechanisms that are energy demanding. I don't know if I'm answering.
Yes, thanks. It's possible if I ask one more question. Sorry, Francesco.
Go.
I was just wondering if anyone has looked at isotope substitution, how isotope substitution changes the creation of biological molecules from abiotic molecules. For example, like an isotopic version of the Miller-Urey experiment or experiments with pyrites.
I think theoretically it would be possible, but the different experiments that have been done with growing animals in media of different isotopic composition different to the isotopic composition of their bodies, or giving animals diets with isotopic composition different of that of their body, they show a resistance to change to make isotopic substitutions, and this is probably related to the fact that each species has evolved with an optimal isotopic composition. So
can I clarify my question? Not even in animals, just when you talked about how abiotic went to biological, they did that experiment where they sent the electricity through the abiotic molecules and made biological molecules. Has anyone tried to do that with different isotopes in those molecules?
I don't think so. I don't know that. No.
Interesting.
But,
Sorry to interrupt.
Yes, because the interaction between isotopes and the influence of isotopes in chemistry, in chemical reaction, I think has never been introduced to biomolecules, or very rarely. There is many work related to the effect of isotopes on enzymatic reactions, but not in other fields of biology, chemistry. So,
Thanks. Thank you.
There's one more question of Omid. Omid, please.
Okay. Okay. Thank you very much for your interesting and unique talk. My question is, in both examples, in the challenge part, you mentioned the calculation is a challenge. I'm curious about why it is computationally or theoretically difficult, or just no one was interested to calculate the theoretical part.
So well, it looks like we have a volunteer.
Yeah.
No, I think it's a challenge, especially because in the case, for example, of the enzyme, it is necessary to know what are the different. It is necessary to know well the energetic profile of the enzyme. So it is necessary to consider the specific thematic steps and how the molecule, the enzyme acts. So I don't think it is theoretically particularly challenging, but it is the challenge is especially coming from the part of the lack of information or a specific information about how these molecules act. This is why I think this is a particular challenge. But I know that you are all prepared to make a very good theoretical calculation. So I don't think it represents a particular challenge for you.
Okay. Thank you very much. So physicists should enter to this field. Thank you.
And Ismael, so it looks like we closed the circle, because when we were chatting before the beginning of your seminar, we were thinking of maybe meeting again next week to discuss a little bit more some technicality, and it looks like there is interest. Yeah, Thomas, do you have an urgent question?
Look, I'm not sure that it is urgent. So if you want to close,
no, I was planning to, but I saw your hand.
Then I thank you so much. I just wanted to ask, I mean, from the two examples, we saw this magnetic field. I mean, the migration feature, which is somehow, the ability to sense magnetic fields, right? And one can imagine that this has that we can use this as in a technology even, right? Sensing of magnetic fields has a lot to do with atoms and in which state we prepare them and so forth. But can you tell me what the enzymatic efficiency or efficiency it was, huh? So the other mechanism that improves the metabolism or gives out more energy. Do you have an idea how we could use that technologically? Could we rebuild that, or what could we do with that?
The field of enzymatic performance and energetics and in technology is mainly related to microbiological devices, because in bacteria that perform enzymatic reaction more efficiently, it could be a technological improvement of, for example, fermentation, which is basically one of these processes. But in the case of the basis of energy metabolism in vertebrates, I don't think that improving or at least studying differences in performance of enzymatic reaction of energy metabolism has been used for any technological application ever. But maybe I'm wrong, and I don't know.
But would it be worthwhile? Do you think it would be worthwhile now to think about using different isotopes to do some energy conversion processes, or you think it's just a biological system?
The effect of isotopes on enzymatic reaction is very well from the classical point of view, but if you introduce the effect of quantum tunneling in enzymatic reactions and add the isotopes to those influence, I'm completely sure that nobody has done a similar thing before. So it's probably an open field.
Thanks.
Okay, then Ismael, thank you very much. It looks like there are lots of open questions that require lots of work.
Yes.
No, thank you very much. This was a really inspiring talk. And I think all of us are looking forward to keep interacting with you and maybe to address some of the questions that you raised this afternoon and that you made us curious about, yeah, because we want to learn more about it. So we will do a little bit of our homeworks by reading all the references that you mentioned, and maybe soon we can meet again, but we can organize it offline and via email to synchronize when we can meet again. Perfect. Then thank you very much, Ismael. Again, usually when people visit us in the pre-COVID times and gave us a talk, we used to invite them for dinner. so we owe you a dinner. Yeah.
Okay.
So let's hope that in the new year, when COVID is more behind us, we can have you in Durban and we can pay out our debts.
Yeah. We will postpone this for the next time after the COVID.
We will make a note of it and we won't forget. Yeah?
Yes. So thank you very much. We will look forward our next meeting. Or
definitely. Yeah. Perfect. Thank you very much. And everybody have a good afternoon, and please stay all safe because COVID is still out there waiting for you. Yeah.
Thank you.
Thank you.
Thank you. Bye.
Bye-bye. Thank you. Thanks very much.
Thank you. Bye-bye.
Thank you. Bye. Bye-bye.